So good morning students. Today we will discuss aerobic respiration. The previous class we have already discussed about glycolysis and fermentation. If you forgot to watch that video, please watch and then join this video. Aerobic respiration. Simple definition of aerobic respiration is you can say that using oxygen and producing energy, the process in which oxygen is utilized and energy is produced. Simple, I will write an equation: C6H12O6 plus oxygen gives CO2 plus H2O plus energy. Let's balance this. 6. So here there should be 6 carbon, 6 CO2. It will come 6 into 8. 8. If I put 6 here, 6, 12. 6 if I put 12, so here I have to put 6, 12, 6, 18, so here I have to put 6 to 18. Like this is the equation. It's a simple equation which you have studied in class 10th, that C6H12O6 plus oxygen gives 6 CO2 plus 6 H2O plus energy. In glycolysis, we saw that glucose is incompletely broken down into 2 molecules of pyruvic acid that we saw. Here in aerobic respiration, we will see the complete breakdown of glucose to the end product, that is carbon dioxide, water and energy that we will get. So glycolysis we have completed. Now glycolysis took place in the cytoplasm. But now the following reactions, that is, there is a connecting reaction called the link reaction, which links glycolysis to Krebs cycle. Krebs cycle is very important. They should be linked, they should be linked. So here comes the role of linked reaction. Let's write about link reaction. Link reaction, very important. For first important topic for this video, link reaction. So we form pyruvic acid. We got pyruvic acid. This we have to somewhat convert it into acetyl coenzyme, acetyl CoA, because that is necessary for Krebs cycle, means it is connecting glycolysis and Krebs cycle. So how we can do? So in SSC or NCRT, you can simply see that here we are doing decarboxylation. What is that? Removal of carbon dioxide. Simple. Removal of carbon dioxide, because this is 3 carbon and this is 2 carbon. This is the first simple step we are going to do. And we will do reduction. We will do oxidation. Oxidation means we will do dehydrogenation. Removal of hydrogen. That is oxidation we are doing. So you know that NAD plus will convert it into NADH. This basic you have the knowledge. Plus S plus H plus we will do. Means we can see here two steps are coming in account: decarboxylation and dehydrogenation. Two steps are coming in account. One more important complex is needed here. What is that? What is that complex? Pyruvate dehydrogenase complex. Pyruvate dehydrogenase. Pyruvate dehydrogenase. Pyruvate dehydrogenase complex. It's a complex which has many that lipoic acid, which we will not study in our course. Lipoic acid is there, B1, vitamin B1 is there. For SSC students, I will tell you one important point, and NCERT students can also refer to it because NEET can frame a question on it. So pyruvate dehydrogenase complex, this we have formed acetyl CoA. Now you can clearly see what we can call this major step as. Many students will call it that it is a connecting step between glycolysis and Krebs cycle. That is correct, very important. 1 mark for you all guys. But can you identify what we can name this reaction? Yes, oxidative. What? Oxidative. Tell me. Oxidative decarboxylation. Very good. Oxidative. Yes, I will move. Oxidative decarboxylation. Decarboxylation. Means oxidation is taking place and decarboxylation, both are taking place in this reaction. This is the first important topic discussed today. Link reaction. Please note down. Please pause the video and note down. Meanwhile, I'm rubbing the board. There's the duster. Now, you will ask me what was the important thing which you are talking about. The important thing is that pyruvate, I will write pyruvate dehydrogenase complex, very important, complex needs vitamin B1, needs vitamin B1 or thiamine, I can call it. Needs because it is very essential. And deficiency of vitamin B1, deficiency of vitamin B1 causes directly they could give you a question that what are the what is caused by deficiency of vitamin B1. So pyruvic acidosis. Second, lactic acidosis. Thereby, it's very important to maintain a proper balanced diet so that this condition is avoided, as these two conditions are life threatening. Now this is done and we got our main product required for Krebs cycle, which many of you by the name of the subtitle came to watch this video. Now is their turn. I will start Krebs cycle. People find it very difficult to learn Krebs cycle. Today I will not complete ETS as I am time bound, but we will complete Krebs cycle clearly. Krebs cycle. Krebs cycle. Why this name? Because of scientist Hans Krebs. He discovered this cycle in the flight muscle of pigeon. In flight muscle of pigeon. In flight muscles of pigeon, and this reaction takes place in the matrix of mitochondria. Matrix of mitochondria. Okay, this is the general name or general introduction. This cycle is also called TCA cycle. TCA cycle. Why this name? Tricarboxylic acid. Tricarboxylic acid cycle. Because citric acid is formed. Citric acid is the first product. That is the first product of this cycle. That's why it is called a TCA cycle, or simply citric acid cycle. Simply citric acid cycle. Now let's move on to steps. We will discuss one by one the steps clearly. Not drawing a flowchart here because it would be quite messy here to understand. So I will deal step by step knowledge. Step by step knowledge. Step 1. Acetyl CoA plus OAA. What is OAA? We have studied in photosynthesis. OAA means oxaloacetic acid, 4 carbon. This is 2 carbon. This is 4 carbon. Determined to make a 6 carbon compound plus water gives citric acid. Gives citric acid. It's a condensation reaction. Condensation reaction. I will write condensation. What is the enzyme? Simple. Citrate synthetase. Citrate synthetase is the enzyme. Now citric acid is formed. Let's move to second step. Citric acid is now. What was next? Citric acid is formed now. Yes, okay. Isomerization, the reaction we will do, and we will form isocitrate, isocitrate using an enzyme aconitase, aconitase, using an enzyme aconitase. Now, third step, isocitrate, we want to convert it into which now? Yes, oxalosuccinate. O sound is coming. Oxalosuccinate. To convert it into oxalosuccinate or oxalosuccinic acid, anyway you can write, to convert this to this. Now we will do dehydrogenation. Simple. NAD plus will convert into NADH. Got it. Dehydrogenation we have completed here. And we got a product, oxalosuccinate we got. So what is the enzyme? Which enzyme is playing role here? Which enzyme? Very easy. Isocitrate dehydrogenase. This simple. Now what I have to do after this? 3 steps are easily understood. 3 steps are like easily clear to you. 3 steps. Okay. Now, 4th step. Pay attention to the 4th step. Oxalosuccinate. I want to convert it into alpha ketoglutarate or glutaric acid, anything you can call alpha ketoglutarate. So I will remove carbon dioxide. That is, I am doing decarboxylation. That is removal of carbon dioxide from oxalosuccinate to give alpha ketoglutarate or alpha ketoglutaric acid, anything you can call. Which enzyme? You only tell me now. Which enzyme will come here? Yes, simple. Oxalosuccinate decarboxylase. Which enzyme? Oxalosuccinate decarboxylase. Now note first these 4 steps, 4 important steps. Pause the video and note these 4 important steps. Yes, now I will rub the wood. Okay. What was the now 4th step? 5th step. Alpha ketoglutarate I want to convert it into succinyl CoA, coenzyme A. So now this is very important step which we are going to perform here. We have to do here removal of carbon dioxide, okay, that is decarboxylation, and dehydrogenation also, that is dehydrogenation, that is I will convert NAD plus to NADH. Now you will think which enzyme should be used. So alpha ketoglutarate decarboxylase dehydrogenase, simple enzyme. Alpha ketoglutarate decarboxylase dehydrogenase. Now you can identify this process is similar to the process of link reaction. This process is similar to the process of link reaction. So similar process to link reaction. Which process is similar to the process of link reaction? Conversion of alpha ketoglutarate to succinyl CoA is similar to link reaction. Let's move on to 6. Succinyl CoA. I want to convert it into succinyl CoA. I want to convert it into succinate. This is also an important process, as for the conversion we have to form GTP from GDP plus PI. We are forming GTP in this process, synthesizing not ATP but similar GTP. They are equivalent. So this process is also called SLP, that is substrate level separate substrate level phosphorylation. That is direct conversion of GDP to GTP, which is equivalent to ATP. So copy the 2 steps, 5th and 6th, and we will move on to the next step. Okay. Now, 7th step. Succinate we got. Succinate we have to convert it into fumaric acid or fumarate. This is also an important process because here we will do dehydrogenation. But here FAD plus is getting converted into FADH2. That is very important. Here NAD is not doing, here FAD is taking a role. So now which enzyme? Yes, succinate dehydrogenase. Dehydrogenase. Okay, now eighth step. Fumaric acid 2 I am converting into malic acid. It is, I can add a enzyme here, that is fumalase, but that is not in the syllabus, so I will keep it as it is a conversion to malic acid. Now this is an important step number 9. It should be a 10-step process, but we are completing it all line by line. You have to note down the processes. Malic acid, we are again converting into OAA using dehydrogenation, that is NAD plus, we are forming NADH plus H plus using mutate dehydrogenase. So OAA, the first substrate we are getting back here. So this whole is Krebs cycle. This process, the whole process is Krebs cycle. Now let's discuss some summarization. I will give you summarization. One Krebs cycle. In one Krebs cycle, you could see one GTP, which I will call ATP. One ATP is synthesized. 4 means 3 NADH are formed. 3 NADH are formed. 1 FADH2 is formed. 2 carbon dioxide are removed. This is taking place. Now for one glucose molecule, we will require 2 Krebs cycle. For 2 Krebs cycle, we will get the product as 2 GTP or ATP, 6 NADH, 2 FADH2 and 4 carbon dioxide. 4 carbon dioxide is removed. This is the energy account for one glucose molecule, 2 Krebs cycles, so into 2 I have done. And further you will study, that is in my next lecture, that 1 NADH gives 3 ATP and 1 FADH2 gives 2 ATP. So we have covered in this video Krebs cycle link reaction also. Krebs cycle plus the link reaction and little bit discussion sort of thing we have done. Now in the next video we will cover important ETS with respiratory balance sheet, and I will try to give you a short video on respiratory quotient. Then this whole chapter is covered in 4 to 3 videos. This is the second video and we are at good pace that we have completed till the end of Krebs cycle. till the end of Krebs cycle. If you have any doubts, you can simply post or DM me, comment directly, and thank you for watching.